proteins made, antibodies made, the cells start dividing. Other leukocyte receptors have ITAMs, and this just goes several of them. There's a variety of them. NK cells have receptors that have ITAMs or co-receptors that have ITAMs. A lot of these have, anytime you see a complex like this, that indicates that that's in the IgG superfamily, that that's a domain like IgG. Some that are important in T cell activation are these B7-1, the B7 receptors. These are adhesion molecules. The B7-1 or the B7-2, and they bind to CD28. That's important in T cell activation. Cytokine receptors can be very similar. They can have cytoplasmic extensions that become phosphorylated, and those in turn can activate other things, other kinases, and in result you have transcription factors made. Okay, I'm skipping over a lot in the book here. Okay. Here are some different types of receptors on immunocompetent cells. I don't want you to memorize these, but just know, a lot of these are dimers. A lot of these are dimers. And tumor necrosis factor receptor, tumor necrosis factor receptor family, there's a whole group of these that are similar. Chemokine receptors, we talked about the CCRs and the CXCR4s, CXCRs. These have similar shapes. So they are studying these receptors in order to understand not only how the signals bind them, which it'd be nice to know that in case you need to block it, right? But how the signals bind them, but also how the transduction occurs. And a lot of the key research being done in immunology right now is in the cytoplasmic signals and how they work. Okay, some things that can happen with certain signals, and this is one of my favorite topics. There are two types of signals I want to make sure you know: the FAS and the tumor necrosis factor receptor. The FAS, shown here, this purplish receptor, and the tumor necrosis factor receptor 1, shown here in blue, have cytoplasmic extensions called death domains. I love that name. Death domains. And you can see the death domain is an extension on this peptide, and when it forms, when it binds the FAS. Now, here's some terminology you need to get used to. This is the FAS. It's an adhesion molecule. It binds another adhesion molecule called the FASL. That L stands for ligand. That's the adhesion molecule that binds the FAS. Two different types of cells. Okay? One type of cell has the FASL, another type of cell has the FAS peptide. Okay? When this complex, when these two adhesion molecules bind, you have the activation of the death domains. This structure, the FADD, binds, and that's an activated structure, and it in turn activates something else called the caspase cascade. This is a procaspase. I'll talk about it in just a minute. On the tumor necrosis factor receptor, the tumor necrosis factor can be an adhesion molecule. It can also be a signal that's secreted by a cell. When it binds this receptor, you have the activation of the death domains. Okay? And the end result of this is that you have transcription factors being done. What happens here is that the FAD and the TRADD also have death domains, and these death domains activate the caspases. Have any of you heard of caspases before? Mixed bag. Caspases are proteases, and in it's kind of like the complement cascade. Remember, we went over it quickly, but the complement proteins, a lot of them, when they became activated, they became a protease, and they clipped to the next protein, and that next protein became a protease that clipped to the next protein. You know, these caspases are protease precursors. They're, you know, the procaspase can be converted into a protease, and that in turn will cleave the next caspase, which cleaves the next one, and so forth. Is that? It's a cascade. This occurs in the cytoplasm. The end result is the caspases activate transcription factors, which result in apoptosis. That's why they call these death domains. The death domains help activate the caspases, which in turn activate transcription factors, which end in the cell dying. This is the extrinsic pathway of apoptosis, of triggering cell death. And so death domains are a good term for those domains. This is the intrinsic pathway of apoptosis, and it can happen when cytochrome C is released from mitochondria. That's a protein that's involved with electron transport, right? If a mitochondrium is damaged, then it can go into can be released into the cytoplasm, and that's going to trigger apoptosis. Apoptosis can also be triggered by UV light, damage to the DNA with thymine dimers, right? Or toxic chemicals, or lack of nutrients, or lack of growth factors. But essentially what happens is that cell is going to die. Now, we're going to talk about the FAS and the FASL and the tumor necrosis factor later, because those are involved with our cytotoxic cells that we've discussed. Toll-like receptor, and that's discussed in another chapter, sorry. I'm not going to go into it now. Toll-like receptors bind to microbial products. Toll-like receptors, we talked about them briefly. They have this C-like shape, and you know that they bind to HAMPs, right? And they have transduction. Transduction occurs with them. You can have a death domain, or you can have something else occurring where you have activation of transcription factors. Okay, that's that chapter. I tried to do that short and sweet. Development of lymphocytes. We have about 10 minutes, and I'm going to try to do what I can with this chapter, so I'm not going to let you out yet. Okay, 10 more minutes. I'll let you out at 3:30. Development of lymphocytes. This is a long chapter. Ayla, do not read this chapter. This is a long chapter. Do not read this chapter. I love to see your smiles when I say that.
I'll tell you to read it.
This goes into a lot of developmental biology. And I want to hit the high point because we need to get on to other things. I want to talk about immunity to disease. I think you're going to be more interested in that than this. Okay, so don't feel like I'm slighting you. You're going to get your tuition in this place. I'm sorry, I know you haven't been worried about that. Lymphocyte development. Okay, how do these B cells and T cells mature? That's what this chapter deals with. How do the B cells and T cells mature? We've already talked about a lot of this. Where do they originate? Bone marrow. Where do the B cells mature? Bone marrow, or the bursa, in a fetus liver, to some degree, they have found. What about T cells? Thymus. They're made in, they originate the bone marrow, mature in the thymus. So you know the fundamentals. I just want to go through and add some more points to that. Okay. Stromal cells are connective tissues that have an abundance of MHC complexes and other adhesion molecules. They are important in, some of these are called nurse cells. They expose those maturing B cells and T cells, primarily the T cells, to self-antigens, to the MHCs. They're important in helping those cells identify self versus non-self MHCs and other adhesion molecules. Does that make sense? They have long extensions like dendritic cells, a lot of surface area. They also secrete cytokines and chemokines that influence the maturation process. So these are critical. And this just goes through a maturation process. B cell development and activation. Let's deal with it first. This occurs in bone marrow, and it shows the femur here. It's also, no, that's the humerus. I'm sorry, femur's in my leg. It shows the humerus and the femur. Also, the sternum is also important in this process. You can see the bone marrow stromal cell right here. Long extensions. Adhesion between the precursor B cell and that stromal cell. During this maturation process, the B cells mature and they start expressing surface antibody. They go, and this shows them still in the bone marrow. There's a little blip here I want to point out in a minute, but they will express surface antibody, and then the surface antibody may disappear on the cells. But when they go into circulation, they have surface antibody. It may be IgM or IgD, as we've discussed. And then activation process, it binds antibody here. Cross-linking, this is a great picture showing cross-linking of surface antibody. And multiple surface antibodies being bound by the same antigen. You have the B cell activation, you have the clonal activation, you have plasma cells, memory cells. What I want to do is briefly go through that whole maturation, activation, development process with major changes that occur within those cells. The things that they can produce and the things that are expressed on their surface. Okay, and I don't want you to memorize all of these. I'm going to try to point out the most important parts. If you look at this, here we start with the stem cell. Okay, this shows the heavy chain genes. Oh, we've talked about this. You know, the V, D, J, C. Here's the light chain genes, V, J, C. You know, the lambda and kappa. And then we have surface antibody. Okay, just with that in context. Stem cell, germline. Here we have, it's starting to differentiate. It's doing some rearranging of that antibody gene, right? We've done some more. The L chains, the heavy chains can be expressed at this point when we've gotten to this point. But please look, you don't have any surface antibody in those previous cells. These immature cells don't have surface antibody. But we get the heavy chain figured out first. And that can be expressed. It's going to have any light chain there? No. And we start having the light chain rearrange, and you don't have expression here until we have the light chain figured out, and then that surface antibody can be exposed. And here you have heavy and light chain expressed on the surface. And it is in that either IgM. IgM is the early antibody. It's made first. And it's going to have IgM when it's in the bone marrow. When it goes into circulation, it can have IgM and or IgD. Does that make sense? So, you know, the only thing really new here is that we have the heavy chain rearrangement done first, and you may have that expressed on the surface, but then you have the light chain and the intact surface antibody is expressed later. Okay. Okay. This goes into different markers on the surface. A lot of these are CD markers. Some of them are transcription factors. Don't worry about them. These are things we talked about with signal transduction, some, Ig alpha, Ig beta, and so forth, and the RAGs and so forth. I didn't go into detail on these, okay? You can see, big picture, during the maturation process, stem cell, early pro, here we have the heavy chain ready, then it's reabsorbed, and here we have the heavy and light chains expressed on the surface, but still in the bone marrow, and then it's in circulation, okay? You see the progression. Please notice you have a difference in the genes that are